much, everybody, for coming. Just get our stuff set up here. All right, guys. Thanks so much for coming. So I'm in the Cell Molecular Biology graduate program, and I'm really excited to talk about my thesis project today with all you guys. So as Patricia just briefly mentioned, I'm going to tell you a story about a certain gene today called cactin. And this gene became of interest to the Maison lab actually before I joined the lab. So they did a screen for proteins that change in abundance in the root tips of seedlings that have been turned on their side. So seedlings can sense which direction gravity is coming from and then send their roots downwards and their shoots upwards. And that process is still not completely understood, how those plants are sensing gravity and how they're redirecting their growth towards it. So this screen was hoped to find new proteins that are involved in that process that we didn't know about previously. So one of those proteins that came up is called cactin, and it's almost been like a wild bull that we've kind of ridden out of the room of gravitropism and into a whole new arena. So I'm really going to talk about where that protein's taken me today, which is actually away from gravitropism and into something entirely different. So it's been an exciting adventure following cactin where it takes us. So the title of my talk today is Cactin is an essential nuclear protein in Arabidopsis and may be associated with the eukaryotic spliceosome. So I'm just going to start with an introduction to this conserved eukaryotic gene in general, and then we'll drill down a little bit deeper in a minute. So here we have our tree of all of life. And when we look for the sequence of the cactin gene in these different organisms that have their genome sequenced, we find this gene present in essentially all eukaryotes. So from this, we would infer that likely this gene came into being probably in the eukaryotic common ancestor. One exception to this is the subphyla that includes Saccharomyces cerevisiae, baker's yeast, has lost this gene, which is kind of interesting. Other than that, we find it retained in all the other genomes I can get access to. So in order to address how well this protein sequence is conserved between these different organisms, how similar is it, I'm showing you a protein dot plot here. So I kind of like this type of graph. It's kind of cute. So if you imagine the sequence of the human cactin protein running along this axis from its N-terminal end to its C-terminal end, and the sequence of the Arabidopsis cactin amino acid sequence running along here. Every place where there's a few amino acids in a row that match each other, you get a dot. So if you dot plotted the same sequence against itself, you get a perfect diagonal line. But here you can see there's some degree of matching along the length of the protein with some patchy regions. But I show this to you just to show that it's not a single domain that's been shared between these different proteins. There's at least some level of matching along its length. I'm now going to show you that amino acid alignment for this region down here. So it would correspond to this part of the human sequence and that part of the plant sequence there. So this is just a truncation of the alignment, and we'll keep looking at it that way, but it's going to include some plants, some fungus, some animals, also toxoplasma, so it's a single-celled pathogen. It's that cat scratch fever. And as you can see in this domain, of course I'm showing you the most well-conserved domain, but in this domain, there's really a nice level of conservation of this protein sequence along the length of cactin. What does that really tell us about what this gene might be doing? So when we look at the different functional domains in this protein, we don't really have any domains of known function. There's a coiled domain here, so it's a super common domain involved in protein-protein interactions. There are nuclear localization signals, and these features are also true for, I'm showing Arabidopsis cactin here, but these features are generally true for most all the other organisms as well. Perhaps one of only the only clue that we get from the amino acid sequence about what this protein might be doing is this region which PFAM predictors come up with as low complexity, probably because there's a lot of arginines and serines in this area. I'm going to talk more about what that might mean a little bit later, but I've got all the arginine shown as a blue line or as a purple line, and all the serines as a purple line there. So this might be our only clue as to what it's doing. Other than that, we don't get much from just looking at its amino acid sequence alone, any clues. Another interesting thing to note is this gene doesn't have paralogs in most organisms, so it's single copy in most all the organisms. What do we know about what it might be doing? So we know that whatever cactin's doing is doing something important. So mutants in this gene in Drosophila die in the larval state. Same with C. elegans, those mutants die as well. A temperature-sensitive mutant in Toxoplasma results in cell cycle arrest. So when you're a single-celled organism and you have a cell cycle arrest, that's also the end of the line for you. And also in zebrafish, morpholino silencing against cactin results in really abnormal zebrafish, and most of the zebrafish die really early. So whatever it's doing, it's important, but it still could be doing just about anything. So at the start of my project, no molecular function for cactin had been proposed. Other than knowing that it was important, that was all we had. There had been a couple of small papers on this subject. They had found some evidence that cactin might be able to change gene expression of many genes, but as to how it was doing that, what was it doing, it was really open book. So it was my goal to try to figure out what is cactin doing. Nobody had looked at cactin in plants yet. Maybe plants are going to be one of the places where we might have an easier time figuring out what this gene does. So we started off with a genetic approach. So there was one mutant allele of cactin available. So this is a large tDNA insertion in the middle of the cactin gene. This is about 5 to 10 kb insert right in the middle there. And when we ordered this seed containing this mutation from the stock center, we found no homozygous seeds in that original batch that they sent us. We did have heterozygous seeds, which we grew up and allowed to self-cross. So you would expect 1/4 of the offspring from a heterozygous self-cross to be homozygous mutant. But we found 0 homozygous mutants. We did get a healthy seed from these plants that germinated essentially fully. So somewhere before the state of being a seed, we were missing our homozygous mutants. So we got 2/3 of the seedlings that we got were heterozygous and 1/3 were wild type. So this is consistent with the 1/4 homozygote just missing from our population. So when we looked at the calyx from these plants and broke them open under a microscope, we could find these small, underdeveloped seeds in the calyx from a heterozygous plant. So calyx are just the seed pods that Arabidopsis put their seeds in, so they look like this. It's kind of like a pea pod. You could think of it that way. So when we counted these small guys in the calyx from a heterozygous plant, we found about 25% of the seeds in the calyx were these small, underdeveloped seeds here, where we find very few of these undeveloped seeds in wild type. So in answer to where are our homozygous mutants, that's right where they are. They're not even making it into being a seed. So we looked at these small, underdeveloped seeds more carefully and under the microscope to see exactly what phase in development are these plants failing to develop. So right now what I'm showing you is just a wild type embryo from a wild type plant that is just 2 days after that flower has opened and then subsequently self-pollinated itself. So at 2 days old, and this would be what will later become the seed, if we looked at that here, and this little guy here is the baby plant embryo. So this is just 1 or 2 cells large at this state, and this is what they normally look like at this age. If we look a day and a half later, they've grown much more, and they now have this globular state that kind of looks like a lollipop. And in the calyx from the heterozygous mutants, we find 75% look like this at this stage, 3.5 days after the flower is open. However, 1/4 of them still look like this. So it looks like our homozygous mutants are making it to like the 1 to 3 cell stage of development and then just stopping right there. So we were once again kind of left with this issue of cactin being a really important gene, but what is it doing? So we can see what is it doing? So much like animals, we found that this mutant was early embryo lethal in Arabidopsis. So another way to try to gain insight as to what this protein might be doing is to see where within the cell is cactin localized to. So I made a 35S, which is a strong ubiquitous promoter that we use a lot in Arabidopsis, to drive the genomic version of cactin. You can see its exons here, and it's got a C-terminal red fluorescent protein tag. And we transformed this into the heterozygous mutant, and in subsequent generations, we were able to find homozygous mutants, meaning that this transgene does successfully rescue the lethal phenotype of the cactin mutation. And these give rise to plants with a normal phenotype. But let's see where we're seeing our RFP tag now. So this is a bright field picture from the mature root, which is kind of just in this upper area of seedlings. And when we look for RFP, we see cactin RFP localizing to nuclei, to nuclei of the root. So here I'm using DAPI, which is a chemical stain that stains both nuclei and the cell wall. Actually, I'm going to switch this here. Is that a little bit better to see the slides? Okay. So you can kind of see that all of our red dots are occurring on top of these blue dots. And so this was showing us that cactin is expressed in the nucleus of these root cells. And in many nuclei, we just see the red staining throughout the nucleus more or less evenly. But in some cells, we see something a little bit different. So in some cells, we see that within the nucleus, we have these speckles, these discrete speckle shapes. When we overlay that with our DAPI staining, you can see these red dots within the nucleus of these cells. Interesting thing about this is not only is cactin nuclear in these other eukaryotic species, but it also forms speckles in at least C. elegans in humans, and in toxoplasma, their microscopy wasn't zoomed in enough to tell one way or the other. So it looks like this localization of cactin to the nucleus and to sometimes as speckles in the nucleus is also conserved throughout eukaryotes. So there are lots of different types of proteins that can form speckles in the nucleus, but one major class of proteins that's known to have this, this localization, and particularly this ability to sometimes be spread throughout the nucleus and to sometimes be speckles, are spliceosomal proteins. So this was one of the first pieces of evidence that we had that made us start thinking about the spliceosome. So spliceosome proteins in animals and in plant cells can make this transition between being widespread throughout the nucleus and just in these discrete speckles. And I want to take a minute to talk about why we think that's true, just based on lots of work that's been done on these spliceosomal proteins in all sorts of organisms. So there's this general trend that the more active a cell is, the more transcription it's doing, the more transcripts it's making, the more splicing it would be needing, the more of a widespread type of localization that we see. Versus if you have a cell that's not doing as much transcription, a little bit more quiescent, those are the cells where you tend to see more of these discrete speckle localizations for spliceosomal proteins in general. So the thought for why that might be true is that in an active cell, you've got all these little spliceosomes more spread throughout the nucleus that are actively working, taking introns out of mRNAs. So in a less active nucleus, they're like hanging out, relaxing, not doing as much active work. And for some reason, they seem to clump together in this inactive state. So there's a lot of correlative evidence that supports this general theory, but it's not completely understood yet. So do we see the same sort of correlation between active cells having a more widespread pattern and inactive cells having a more speckle pattern with cactin? So here I'm showing you different cells within the mature root region of a seedling, and particularly in younger seedlings that are much smaller, I see most of the cells looking more in this diffuse widespread type of localization. As the seedlings get older, I start to see more of a transition towards these discrete speckles. When I look in the root meristem, so it's located at the tip of the root there. So there's a lot going on here. These cells are actively dividing. You can see they're much smaller cells. So each cell would be about like this. They're much smaller cells. In this region, they're actively dividing, actively differentiating. We would expect them to be very active. And I always see a more widespread localization for cactin in those cells. The hypocotyl, which is kind of like the stem of the seedling, I tend to see these larger speckles within the nuclei. Those cells have smaller nuclei. And in leaf cells, I kind of see this intermediate state where it's not as discrete of speckles as in some of the other cells. One thing you might be wondering is these constructs I had shown you thus far were all done with this 35S very strong ubiquitous promoter we use in the lab a lot. But I also made a construct using cactin's native promoter, as well as its native 3' untranslated region and 5' untranslated region, also with a C-terminal RFP tag in it. And when I transformed this into plants and looked at them on the microscope, I see the exact same patterns. So I don't think these patterns I'm seeing are an artifact of the promoter. So those kind of correlative things were interesting, but it made me wonder, can I actively perturb the localization of cactin? If I actively make a cell become more inactive, can I make cactin change from being more widespread to being more speckled? So here what I'm showing you are cells from the mature root of a very young seedling. So I tend to see more of the widespread pattern of cactin. So you can see these are 3 nuclei here. These are just example ones. Here's another one. This one has some speckles in it and also some kind of staining throughout the nucleus. So while this seedling was still on the microscope, I squirted under the cover slip some sodium azide. So that's a potent inhibitor of respiration. So you are going to be starving these cells. So you would expect a lot translation and then transcription to kind of shut down to conserve energy. And so you'd expect there to be a lot less splicing needed in these cells after the sodium azide is added. And then these are those same nuclei photographed just a couple of minutes later. And I see this transition within a minute at the concentration of sodium azide that I was using. And so these same nuclei do indeed become much tighter speckles after I add the sodium azide. So another thing you might be wondering is, what does sodium azide do to these transgenic fluorescent proteins in general? Is it possible that sodium azide just causes these transgenic proteins to clump together? So I was able to get some phytochrome B GFP seeds from the Virster lab next door, which was very nice of them, to use as a negative control. So phytochrome B is an Arabidopsis protein involved in light sensing and the shade response. But like cactin, it's a nuclear protein. And depending on the treatment that you give the seedlings, it can be either diffusely stained throughout the nucleus or on these more discrete speckles. However, it has not been implicated in splicing, and we don't think phytochrome B is part of the spliceosome, as best we know. So it's kind of a nice negative control for another protein found in the nucleus, tagged with the fluorescent protein, that we would not expect to respond in the same way as spliceosomal proteins. So for this experiment here, I grew them completely in the dark so that the phytochrome B GFP would be kind of widespread throughout the nucleus. I added the sodium azide to see if that would make it go into speckles. And as you would hope for a negative control, it did not. So after adding sodium azide to these seedlings, that phytochrome B just stays in the nucleus, kind of widespread. So it shows that we're not just getting clumping of these overexpressed fluorescent proteins. This looks like, maybe a more specific response that cactin was showing. So this change between being more widespread and more speckled after the cell was put into a starvation state really fits with cactin, maybe playing a role in the spliceosome. So next thing we wanted to see is, are these speckles that we're seeing the same speckles that known parts of the spliceosome localize to? So I'm starting off here showing you some cells that don't have speckles. So these are in that root meristem again. So you can see each of these are very small cells, and we always see cactin showing a more widespread localization in those cells. And this next protein I'm going to show you is called SR45. So this is a known component of the spliceosome. It's named SR because it has a domain that almost goes arginine, serine, arginine, serine, arginine, serine. And so that's what the S and the R is for. And many of these spliceosomal proteins have a domain that is very rich in arginines and serines. And that's thought to aid in all the protein-protein interactions necessary to build the spliceosome. The spliceosome has over 300 different proteins in it, so they really need to bind together. And that's what this domain is thought to be important for. As I mentioned before, cactin might have a weak domain that's a serine-arginine domain. But getting back to SR45, we see, just like cactin, it forms these more widespread localization within the nuclei of these very active cells in the meristem. When we move up to the mature root region, where I tend to see more of these speckled cells, we find that indeed cactin is making these same speckles as SR45s. You can see that on the merge here. I used another known spliceosome component as well, called RSP31. And again, it's got that name RS because it also has an arginine serine rich domain. And I again see the same speckles there. Kind of nice thing about this protein is its GFP fusion protein has been confirmed as the same as antibodies against the native RSP31 locus. So it kind of shows that even overexpressed with a GFP tag, this is the natural localization for this protein. The antibody is not available anymore, but it kind of legitimizes that as a speckle, a splicing speckle marker. So again, I wanted to use a negative control for this, because these are all overexpressed proteins with these fluorescent tags on them. Is there any chance that they're just clumping together because they're overexpressed, and that's why they're all colocalizing? So again, I used phytochrome B GFP as a control. Because I was interested in if we're getting the same speckles, I this time treated them with the 2 hours of light treatment in order to have it form speckles. And what I find is, although in the cells I was looking at, cactin is in speckles and phytochrome B is in speckles, it's not the same speckles. So these are not just the speckles that all overexpressed fluorescent proteins are forming. So I was just able to show you a couple here in the PowerPoint, but we looked at many more nuclei, and we quantified how well the RFP and GFP channels were overlapping. So this is done by looking at the correlation of coefficient between the pixel intensity on the green channel and the red channel. So a perfect correlation would be 1. And you can see that cactin RFP with GFP SR45 and cactin RFP with RSP31 both give an extremely high level of correlation between their localization. Our negative control down here comes in much lower, and it's statistically different from our proteins of interest. So this was showing us that the speckles that cactin was localizing to are the same speckles that these spliceosomal components are localizing to. So this was also fitting with our new hypothesis. So we took another approach to try to figuring out what cactin does, and this is an unbiased screen, so anything could have come up. So this was a yeast-2 hybrid screen where we're looking for other proteins that cactin might bind to. So we sent off our cactin plasmid off to the Indiana University's yeast-2 hybrid facility, which I highly recommend. And they screened in Arabidopsis seedling cDNA library as our prey library. The vast majority of the colonies that we got from this screen were a previously unnamed protein that we started calling rintin for relationship to cactin. And I'm just going to show you our yeast-2 hybrid conformation real quick of this screen result. So each row is a combination of our 2 different genes showing which vector that they're in. So this second panel is mostly the same, but everybody's switched as to whether they're in the DNA binding domain vector or the activation domain vector. And then I'm showing 2 transformant colonies from each combination in a 10-fold serial dilution for each of them. And this first whole half is on the media selecting for an interaction, versus this whole half is just a control, not selecting for an interaction between the proteins. So these here in yellow are all negative controls. So it's cactin with some autophagy proteins, which we were again kindly given as controls from the Virster lab next door. So these are just autophagy proteins that, as I'll show you in a minute, are expressed well in the yeast, and yet it's just showing that cactin alone cannot auto-activate this assay, same with rintin. So we have our negative controls here. Then as a positive control to show that our assay is working, we have these 2 autophagy proteins together, which are known to interact. And as you can see, they grow nicely on the selectable media, as you would expect. And here's our experimental control. So cactin and rintin, no matter which one is in which vector, they interact with each other and grow on the selectable media. So what is rintin? So a lot like cactin, rintin is conserved throughout eukaryotes and contains no domains of known function. The one domain it's recognized as having is domain of unknown function, number 167. It's a pretty small protein. And what really got us excited about this result was we found from high throughput literature on Drosophila, where they had done thousands of yeast-2 hybrid screens on thousands of different proteins in Drosophila. Drosophila cactin was found to bind to Drosophila rintin. So this is suggesting that this pairwise relationship between cactin and rintin is conserved between plants and animals as well. So when we started trying to find out everything we could about rintin, one of the few things we could find that is known about it is that rintin has come down in screens for being a spliceosome-related protein in both human and Drosophila cells. So it's looking like we've got cactin showing a conserved relationship to a protein that at least in 2 different screens has been shown to be associated with the spliceosome. So this was another strong piece of information that was tying cactin to the spliceosome. So we wanted to see, where does rentin localize to? So if rentin and cactin are genuinely interacting with each other, we'd expect them to be in the same part of the cell so that they really could interact with each other. So what we find is, much like cactin, in the meristem of the root, we find rentin in this more widespread localization. And in the mature root, we also find it forming these speckles. And when I cross these plants to plants with RFP cactin, I found once again it's the same speckles. So these 2 proteins are co-localizing with each other. And we quantified this as well. And so you can see a very high level of co-localization between cactin and rentin. So they're in the same place at the same time. So rentin is what I would consider single copy in Arabidopsis and most of the other organisms, but it's a little bit complex, so I wanted to explain. So it does have a distant paralog that we find in these different organisms, which we're calling syntin, because even though rentin's a pretty short protein, syntin is even shorter. So for shorter rentin, we called it syntin. And when we make a phylogeny of all these rentin and syntin genes from plants and animals, we find all the longer rentin proteins clustering together and all the shorter syntin proteins clustering together. You might notice that fungi are prominently missing from this phylogeny. And it's because, kind of like cactin, this protein has been lost in different times within fungi. It's been lost more than cactin has, actually. So this phylogeny with all of these clustering together and all of these clustering together would suggest that the eukaryotic common ancestor had both a rentin gene and a syntin gene, and that these organisms that have descended from the eukaryotic common ancestor have been maintaining both of them. So our best bet would be that they probably have at least somewhat different functions for them to both to be maintained for so long, but perhaps a related function. I think it's worth noting also that prokaryotes have a domain of unknown function number 167 as well, including E. coli, but we also don't know what it's doing, what those domain family members are doing there either. So once again, we took a genetics approach to try to figure out what rentin is doing. So we looked at 2 alleles of rentin, so we called them rentin-1 and rentin-2, and 1 allele of syntin. And so these are, again, large tDNA insertional mutants, so 5 to 10 kb or so large inserts in the middle of these genes. And we think that these do disrupt the gene pretty effectively. So I did PCR on the cDNA to find that out. And the amplicons that I had are described as these little lines here. That's dotted line to go through the intron. Since we were doing PCR on the cDNA, there was no intron at that place. But blue means we did get a PCR product, and gray means that we did not. So you can see reaction 1, 2, 3, and 4 here for rentin, for example. Same thing for syntin. However, there we just get products downstream of the insert. And I'll just real quickly show you how that's done. So here are some gels from this PCR on the cDNA. And I'll just show you this example with rentin up here. Here's a negative control for contaminating genomic DNA or any other contaminants. And this first gel is for a reference gene, so it's a totally different gene to show that we are actually have cDNA in our tubes. And then this is that reaction 1, 2, 3, and 4 that you saw in the cartoon. For wild type, the wild type plants are expressing full-length rentin protein, so full-length rentin cDNAs, so we get bands for all the wild type plants. However, when we look at the rentin-1 mutants in this example, we find we do get a band for reaction 1, but not 2, 3, or 4. So these mutations are having a large effect on the mRNA, so you would hope that these might be knockouts or at least severe truncations of these proteins. However, much to my surprise anyway, I found that these seedlings grow totally fine. So they have no abnormal phenotype under my growth conditions, So I have here wild type, rintin-1, rintin-2, syntin-1, and a rintin-syntin double mutant, in case they did have some overlapping functions. And at least under our perhaps somewhat idyllic growth conditions in the lab, they all grow just fine and show no abnormal phenotypes. So these genes do not appear to be required, as best we can tell, under our growth conditions. So when we started, based on those other results that I mentioned, digging into the spliceosome as deep as we could to try to see what cactin might be doing, we found one paper where cactin itself had pulled down with the spliceosome in human cells, also suggesting that cactin might be involved with this. We also started looking at any of the protein-protein interaction databases and high throughput work that's been done in other labs. And this is a compilation of lots of different papers that I pulled together for this. But each of these boxes is a protein, and the color of the outline represents which organisms for which I can find a good ortholog for that protein. So all of these have good orthologs in all three of these organisms except for I-kappa B-like. So here we have the connection between cactin and rintin that I found, and then also that one from the Drosophila literature that I told you about. But we also find these connections from the literature between cactin and SR45, which in human cells is called RMPS1. And so SR45, as you recall, is that protein I showed cactin colocalizing with in my plants. We also find a connection between cactin and SMB, which is one of the absolute core components of the spliceosome. As I just mentioned, that one direct connection between cactin and the spliceosome. Pinin is a really interesting protein. So in animal cells, it definitely has a role in the spliceosome and also has a role on the periphery of the cell in cell-cell adhesion. We do find a distant ortholog of this gene in plants that nobody's looked at yet. It would almost certainly just have the spliceosomal role if it was going to share anything with its animal counterpart, because the type of cell-cell adhesion is totally different in animals. But that would be really interesting to look at and see what that protein's doing in plants. I-kappa B, don't be misled by the name, is not really that much like I-kappa B, but it's a nuclear protein that there's a number of lines of evidence connecting it to the spliceosome as well. So we've kind of got this network of interactions that are kind of supporting this connection to the spliceosome. So since I had my yeast-2 hybrid system up and running, I checked for pairwise interactions between cactin and these different components. And it's important to note that most of the data that I took from the literature in order to make this diagram was from immunoprecipitation-based experiments where there might be many proteins involved in these interactions, versus the experiment that I was doing in yeast was just pairwise. And so it had to be that the two proteins I put in the yeast together were sufficient for that interaction. And I did not find positive results for cactin with any of the other proteins listed in this diagram, and I also tried some other ones as well. So, and I think it seems reasonable that this might not be just a pairwise interaction. It might be that it requires its host of proteins in order to have these binding relationships. But I still think this network does draw yet another connection between cactin and the spliceosome. So I want to summarize all those connections that I've talked about here today. So first of all, we have that cactin might have a weak arginine-serine-rich domain, which is a common feature of spliceosomal proteins. Cactin shows a dynamic localization between being more widespread versus more speckle, and we can perturb that localization with sodium azide. Cactin co-localizes to the same speckles with known spliceosomal components. Cactin has a conserved binding relationship with a protein that might be a putative component of the spliceosome also. And so based on all this, we are proposing that cactin is associated with the spliceosome. As to whether it has an active role in mRNA splicing, or is it riding the spliceosome to get onto mRNAs to maybe affect them in some other way, we really can't say yet. But we now have a hypothesis that we can begin testing for what cactin is really doing. So I want to thank everybody. I want to thank all of you guys for coming, which is great. I also really want to thank all my lab mates, particularly Allison, who's been helping edit everything that I've written in the lab since she's been there, and just a really great friend and helpful in every way. And Satya and Shihang and Amy and Nicole and Richard. We've had really some great undergraduates and some students in the lab right now, including Tanner and Rachel and Caitlin. I also really want to thank the Virsha Lab. As you heard me mention a couple of times in my talk, they've been really helpful with providing me with negative and positive controls for my experiments, and they've also really been good friends in the lab next door. I have a really great thesis committee, so I want to point them out here. We've been funded by National Science Foundation, and also a Hatch grant. And I've also gotten a tremendous amount of help and materials for these people down here. So Donna Fernandez helped me with that microscopy showing the early embryo arrest for my cactin mutant. I've received cactin-related plasmids and materials from Erin Cram, who works on cactin in C. elegans at Northeastern University in Boston, and Ruth Stewart, who works on cactin in Drosophila, who sent me some really helpful materials. And I want to thank Scott Michaels, running that Yeast 2 hybrid facility. That was a really great breakthrough for this project. And I want to say a special thank you to Lizzie, who's an undergraduate student who's worked a tremendous amount on this project with me these last couple of years. And she's now taking part in a very prestigious pre-dental program in Minnesota right now, but we're videotaping this to send her because she was so sad she was missing it. But she's not only a really motivated worker and really smart, but she's also just like a sunbeam to have around the lab. And I want to really thank Patrick, who I think is just the best boss in the world. There's nobody that I would rather work for. I think that, he's unbelievably curious about science. I'm not sure I've ever had a result that he didn't think was totally interesting, no matter what that result was. But I think there's at least one thing that he cares about more than science, and that's us, his lab members. Maybe even sometimes to his own detriment, he is always putting our interest in front of his, and there's really nobody I'd rather work for. He's really a wonderful person. Not just in the lab, but I find myself in tricky situations and, with my condo association, thinking what would Patrick do? And I also would like to thank all my friends and family who've been really supportive throughout the course of my entire graduate career and this project in particular. And I do want to say one thing about what I'm going on to do next, or at least hoping to do next. So I'm going to be putting out my shingle as doing scientific visual communication. So if you want to get some idea of the sort of things that I can help people with, I invite you to check out my website so you can get some idea of the sort of stuff that I can do. But if you guys know anybody who is trying to communicate complex scientific things to any audience, I might be able to help you do that. So I'm fast, I'm very friendly, I don't cost that much at all. So tell your friends and colleagues, and I left business cards at both exits. So so I'm happy to take questions.